Hi guys, my name is Alyssa and I studied abroad in Zaragoza, Spain. I am double majoring in biology and Spanish, which was more categorized as biotechnology in Zaragoza. The first semester I took four classes too, which were pharmacology and environmental science, which definitely helped better prepare me for the internship. I worked second semester. I worked in a lab on campus under the Department of Biotechnology, and the title of my investigation is The Role of Furby Proteins from Anabaena species PCC 7120 on different cellular processes and its effect on canine gastrointestinal health. And directly in the lab, we looked at the role of Furby proteins from Anabaena species PCC 7120, but I am planning on applying to veterinary school this upcoming spring, so I decided to take the research a step further and connect it to the gastrointestinal health of dogs. So what we did was look at different regulatory promoters, and we did protein purification, and we ran a bunch of gel electrophoresis tests to see which promoters binded best to the protein. And what that told us was which regulatory promoter could best help the metal zinc be assimilated by the species of cyanobacteria from the environment, which means that dogs would have less chance, or we would have at least more information on why dogs are getting sick from drinking or eating contaminated substances. So, for example, dirty pond water and things like that. And they're getting sick from the toxins that the metal zinc carries with them. So this picture on the right here is the view from my balcony in Zaragoza, just to give you guys a sneak peek if you are interested in studying there. And here are some pictures I took from the lab. On the left is just like what I saw on the day-to-day. Here are some bottles that were the substances and compounds that I needed to actually make the gels to later load them up with the genetic material. And here, in the middle are the two gels, just some examples. So the first one, you can see that it kind of blends into each other and you can't really analyze the the bands so well. And that was because one of the ingredients, so to say, that we needed to make the gels was either expired or contaminated with something else in the lab, which happens a lot. So we had to run a lot of different gels and isolate different variables to figure out which one it was. And that was another cool part of working in the lab with graduate students, is that they knew the reason behind every step of a process, and they could tweak the slightest thing to get a different result. So that was really interesting. And then the bottom one was when we had finally resolved that issue, and the bands are a lot more clear. And yeah, these are examples of some gels that I ran. This photo is just agar and some petri dishes cooling off and solidifying so that we could later streak them with different strains of the cyanobacterium species, um, to see if they were contaminated as well, which was pretty frequent. And if they were contaminated, then that would skew the result of the gels. So we made sure that they were exactly the strains we were looking for. And here are some photos on or around campus. I think the upper left-hand corner one I took maybe the first day stepping foot on campus. I just thought it was really cool. And then the other photos are on campus. The middle one, they have a lot of cool street art, and this one in particular I liked because it was for feminist movement. And the right photo, I saw on a day-to-day basis on my way to the lab. And my mentor and our peers present their findings at scientific conferences. So I thought that was really interesting to be able to contribute in some way to their findings, whether trial and error or something that actually worked out in our investigation. and I definitely learned a lot from it. And I hope that it can help me in my endeavors on my way to veterinary school. I definitely enjoy working in a lab, so maybe that's something that I'll pursue. Um, and it was really exciting to be a part of this research, especially because the university itself has a history of investigating for proteins. So to be able to look into a specific type was really interesting, and I got to read different papers that they had written. So yeah, I hope you guys can make it to my presentation. Bye.